So what exactly is the chromosome and how does it form? What's the structure of the chromosome? Let's take a selfie. So here we have a schematic diagram of the structure of the chromosome. This is the DNA molecule, the double helix. It's about 10 nanometers in length, okay? 10 nanometers. Okay, so now how does this 10 nanometers in length molecule fit inside of a tiny cell's nucleus? Remember, DNA is found in the nucleus, but not only in the nucleus. We also have DNA in the mitochondria and we have DNA in the chloroplast for plants. So let's come back again. The DNA molecule is long, and in order to fit inside of that tiny cell, it has to coil and supercoil itself to condense in such a way that it will be smaller to fit inside of the cell. Here we also have a practical. So let's say this is the DNA molecule. See how long it is. See how long it is. So it has to coil and supercoil around special proteins called histones. Histones. Now DNA is negatively charged, while histones are positively charged. Remember the law of physics, negative and positive attract. So these histone proteins are positively charged, so they bind to the DNA molecule to form what we call strings on thread. The chromatin structure, just like strings on threads, okay? So it keeps coiling on those molecules to form strings on thread. But then that is not enough. It has to compact even further. It has to coil even further to form the chromatin fiber, as we can see it here. And then that is not even enough. It keeps coiling and supercoiling and further looping, and then to form this structure here, what we call the chromosome. So chromosome is made up of DNA molecule, histone proteins. Okay, DNA molecule and histone proteins compact, coiled and supercoiled together to condense in this form. So now in this form, it will be easy for this particular structure to fit inside of the tiny cell. How can a 2 meters long molecule, if we stretched out all of the DNA inside of the human cell, is about 2 meters. So how can a 2 meters long molecule fit inside of this tiny cell, this nucleus of about 10 micrometers in diameter size? So it is only possible through this process of DNA condensing into the chromosome format. So remember that humans, we have about 46 of these chromosomes inside of our cell. We can also look at another example here. Let's say you want to travel, right? You have a lot of clothes that you want to pack inside of your tiny box. So how can you put all of your clothes inside that small box that you want to travel with? You have a lot of clothes, but you just have one box. So what do? You have to fold them. You fold all the clothes, right? Fold them in such a way that the whole of your clothes, or at least the majority of your clothes, can fit inside of that small box. So that is the same thing that happens here. So this DNA and histones complex is all about a kind of folding structure, a kind of scaffolding. Now this is like a kind of scaffolding process that allows the DNA molecule that is so long to be condensed and compact in this form called the chromosome, so that it can fit inside of the cell.